So greetings, everybody. Thank you for your attendance. We are here in Kenya with very rainy weather. Today it's our last seminar of this series, and we have the pleasure to have two great speakers. Dr. Rodomiro Ortiz from SLU Sweden, which no presentations are really needed due to his extensive experience, and Maria Laura from CONICET Inter Argentina. However, I think I should present Dr. Rodomiro very briefly, what was extremely difficult due to his extensive CV, but I will try my best. And so currently he works as a chair professor of genetics and plant breeding at the Swedish University of Agricultural Sciences, but he was already the director for genetic resources and enhancement program at EiB. Also, several director positions in IITA, for example, the director for crop improvement division, but also director of intensive agroecosystems program for CIMIT, the deputy director general research at ICARDA, and he served as a part-time science advisor of the interim consortium office of the CGIAR. So these are just few of his extensive experience and knowledge. He won numerous prestigious prizes, and still he made the time to accept our request to be here. So Dr. Rodomiro, we are very honored with your presence and time, and I will let you add something more about my words and to present as Maria. Thank you very much.
Thank you, Ana Luisa. Just a rato, I was not Deputy Director General at ICARDA. I was at IITA. I was advisor or senior advisor of the Office of the Director General and Deputy Director General at ICARDA. I'm going to share now my screen. I think, okay. I need to share it first. Not working this. Let's see, sharing. Now you see it? Yeah. Okay. Yes, today we have a talk on the implementation of the excellence in breeding mid-density genotyping for unlocking genetic diversity of an Argentinian public temperate maize collection. And our speaker will be Dr. Maria Federico. But before giving the floor to Maria, I would like just to always remind ourselves where we are here, since we are engaged on trying to make a better world. And for that, we need to accomplish what we have agreed in the UN Agenda 2030, the Sustainable Development Goals. And plant breeding, as you see by the arrows, contributes to several of them, perhaps being the most important, zero hunger. That is, end hunger, achieve food security, and improve nutrition and promote sustainable agriculture. I just taking some couple of slides just to show how is the challenge for us. Because one thing is to say zero hunger, yes, we contribute, but we need to be precise. So we need to go to the target indicators of that zero hunger that are relevant to crop improvement. And I did that exercise, and you see the year, and how then we target products, like for example, to end hunger, we need to have high yielding cultivars, to end all forms of malnutrition, then to improve the nutritional quality, to double the agricultural productivity, then we need to increase the genetic gains. To ensure sustainable food production, we need to have climate resilient, resource efficient cultivars that adapt to global warming and produce more with less. And also maintain the genetic diversity of seeds. And that not only is keeping our seeds in cold storage, but also making available the diversity that is characterized and evaluated so can be used. And for that, we have with the advances in the recent years, several different methods and tools. I'm not going to go through them. And I suppose this will be shared with all of you so you can read later. And what we are seeing is also a change on the way that we have been doing plant breeding. and will continue from what we do today and what will happen in the future. Now we are moving more and more into genomic prediction, leaving behind phenotypic screening. That is still important because it's part of the information we need to include in genomic prediction. We need to select parents now on breeding values rather than on pedigree or just by progeny testing. Speed breeding is also accelerating the way we do our business. No, multi-site testing over years will remain, but will be more focused, more characterized environments. Backcrossing is interesting, but we have today also gene editing, crossbreeding. Using crossbreeding marker-assisted selection is now being replaced more and more by genomic prediction. And the way that in the future we may envisage seeing changes in the way we release cultivars, no? And the role that will be not only on the cultivar themselves, but also on the information they bring. No? And the other will be the change in the way that we produce and sell seeds. No? So those are things that I just wanted to mention briefly before introducing further topic. I think we're going to talk about the excellence in breeding mid-density genotyping for maize. I took this information from the website. No, I think we're talking about 3,300 markers, no, that were developed from breeding lines and land races from Latin America, Africa, and Asia. And based on whole genome resequencing, genotype by sequencing, direct tax genotyping, and map tree, and some competitive allele specific PCR in SNPs. We have finished with a platform that could be useful for breeding programs that are targeting maize. No? The average marker density of this panel is about 1 SNP per 0.72 MVPs. So now I have the pleasure to introduce Dr. Maria Federico. And you see that badger with the chair of Wisconsin is because her and I went to Madison to do our PhD, although with some, I think, more than a couple of decades of difference between the time that she was and I was, since I'm older. But Maria has done a career that has brought her not only from Argentina to the US, but also before going back to Argentina, she went to Chile, and she has been working in different areas of plant breeding or molecular plant breeding, as she preferred to put on her profile, with expertise on plant transformation, double haploproduction, trait development, trait characterization, and trait integration. So I will not take more time. I think that we are here this afternoon to hear Maria. So Maria, the floor is yours.
Thank you, Ramiro, for the kind introduction, and Anna for the invitation. Let me share the screen. I guess. I will start. So I will be presenting, good morning, everyone, and good afternoon for people on Nairobi time. I will presenting, like Rodomiro said, the implementation of the excellence in breeding mid-density genotyping for unlocking the genetic diversity of an Argentinian public temperate maize collection. It doesn't seem to be... Oh, now. I will briefly, let me see, because I need to... Now. I will briefly share the outline of my talk. I will first introduce tell you a little bit about the maize production in Argentina. I'll talk a bit about the history of INTA's breeding program, and I'll tell you a few things about the panel we genotyped. I will also share with you the objectives, materials and methods and results, and would like to share also my conclusions and open up the discussion at the end and receive feedback from you. Argentina is located in the southern half of South America and is the world's 8th largest country, covering an area of 2.8 million square kilometers. This vast and diverse territory expands across a spectrum of climatic zones and from tropical to subantarctic. Argentina is a major agricultural producer, well known for its beef and wine industry, but also a top producer and exporter of maize, wheat, soybeans, lemons, among other products. Therefore, Argentina contributes to global food security through agricultural exports. If we focus specifically, if we focus on maize, Argentina is the 4th global maize producer, behind giants like the US, China, and Brazil. Depending on the year, Argentina could be the 2nd or 3rd exporter behind the US and sometimes Brazil. It really depends on the season. And it is capable of supplying external demands of other countries. For example, basically, Argentina supplies 22% of Asia external demands of corn, of maize. It also exports to South America and Africa. Public maize breeding in Argentina dates back to more than a century ago. At the beginning, the objective was very simple and clear. The Ministry of Agriculture wanted to improve yield and help growers financially by providing them with better materials to grow. The result was a release. The first hybrid to be released from the public sector was in 1949. And INTA, the institution where I am based at, the National Institute of Agricultural Technology, was created or founded in 1956. It was then when the maize breeding took a boost, and maize breeding, got more finance and resources and more people, professionals working towards improving maize in Argentina. In 1969, a germplasm bank was created, and that was very important for collecting land races and receiving seeds from other regions. And for 20 years, the program worked really hard in the development of orange flint lines, which are the characteristic lines, maize lines in these regions, creating open pollinated varieties and double cross hybrids, which laid the foundation for both public and private breeding programs. In 1992, a milestone was the establishment of a consortium with private seed companies, which with the objective of maintaining genetic diversity and improving lines and populations. Doing the research local research that seed companies may not be eager to do, and exchanging materials with INTA. The most common breeding strategies followed to develop hybrids in Argentina. You can exploit the Argentine orange flint by U.S. yellow dent cross, or the well-known stiff stock synthetic by non-stiff stock. These two crosses generate hybrids. The flint dent hybrid has great abiotic and biotic resistance, and it's suitable for the dry milling industry. Whereas the dent hybrid, dent by dent, has good performance in Argentina, and this has been improving throughout the decades, and it's suitable for the wet milling industry. Flint by flint hybrids depend on the improvement of local flint inbred lines for this purpose, and this has been an ongoing effort. The INTA maize breeding program has a national presence throughout the country. It possesses 5 stations. Yeah? Oh, okay. So I heard something. Oh, okay, again. Like I said, INTA's maize breeding program has a national presence. Germplasm development is performed in 5 different stations. We are located in INTA Pergamino, which is the reference centers for maize breeding, and we also INTA also has different locations, 7 different locations for germplasm evaluation, and there are 2 biotech laboratories that support the breeding, and we also have the germplasm bank in Pergamino as well. Just to tell you a little bit about the temperate inbred maize lines that we have collected or selected for creating this panel. Right now, the panel consists of more than 500 public inbred lines, mostly adapted to the temperate climate conditions. They were developed by intermating open pollinated populations, mainly composites and synthetics, and also some of them were derived from commercial hybrids. This panel encompasses high variation for grain color and texture, maturity, yield, and pathogen resistance. To illustrate the diversity, I'm going to just show you a few results from a PhD dissertation that it's currently going on. This is Ignacio Torrent's PhD dissertation. Ignacio has evaluated 484 inbred lines of this panel for 3 relevant diseases in Argentina during 3 years in replicated trials. I mean, for 3 years in replicated trials. The diseases are mal de Rio Cuarto, which is a viral disease that affects, when it does hit, it affects severely the production of corn, and the well-known northern corn leaf blight and common rust. Ignacio shared with me these results to illustrate the genetic variability present, the genetic diversity present in this panel. we can see here that the for the 3 diseases, there's variation in the disease response of these 484 lines, and that there's interesting lines with resistance for each one of these diseases. There were throughout the years, we different teams in INTA tried to characterize the inbred lines developed by the program, with different numbers of breeding lines being genotype, and also with different technologies, different number of markers. A first attempt to characterizing the lines was done at the biotech lab in Pergamino with the using microsatellite markers, and a second attempt to characterize an effort to characterize the lines was done through the consortium, and 191 lines were genotyped with the maize SNP 50 bed chip. Just gonna briefly tell you that when the lines were 111 lines were genotyped, but only 70 lines were assigned to four subpopulations. And just to describe them, and for you guys to see the variety in origins of the panel of lines. One of the groups was an Argentine by Caribbean derived stock. These are flint lines with variable or unknown combining ability. Another group of lines which were also orange flints, were mainly derived from an Argentine landrace named P465. Which had good combining ability. All these lines have good combining ability with US dents. And there was a third group, basically of the Iowa stiff stock synthetic background, and these lines were dent or semi-dent. And a fourth group of local inbreds derived from commercial hybrids with flint combining ability. So having access to the excellence in breeding mid-density SNP panel let us include more lines not previously genotyped. Remember that I was talking to you about a panel of 500 lines, and also unify marker sets. For instance, the SNP, the Illumina chip is no longer, as far as I know, is no longer available. So we wanted to unify and also do so in a cost-effective manner. The first, we were able to genotype a first set of lines. For those of you who use this panel, you know that you can only send 4 plates at a time. So, I mean multiples of 4. So the first set of 4 plates carried 376 inbred lines in them, and those lines were genotyped a year ago, and remaining lines will hopefully be genotyped soon. We believe that the future implementation, these data will let us implement genomic prediction and genomic assisted breeding that we will use for germplasm organization and trait integration. So the objective of the study, what I'm going to show you, was basically to assess the genetic diversity, the degree of linkage disequilibrium, and the genetic structure of an extended panel of temperate lines. I'm saying extended because previously we had only a genotype up to 100, up to almost 200 lines, with the ultimate goal of organizing the program through data-driven decisions and hopefully establishing, I mean, this is a long goal, a long-term goal, establishing heterotic patterns, and obviously, like any breeding program, exploiting and maintaining genetic diversity. So we collected collected leaf punches from 10-day-old seedlings. We did it for 484 temperate inbred lines, even though we only sent 376 for genotyping. These 484 lines are the ones that have been phenotyped for the 3 diseases. Ignacio will hopefully model some genomic prediction models for this with this phenotypic data and genotypic data. So freeze-dried samples were sent to Intertech Australia for DNA extraction and genotyping with the 3.3K maize dark tag SNP panel. We used ASR Genomics and SRIP Ready to filter and analyze the genotype data we received back from Intertech Australia. We used Tassel for kinship linkage disequilibrium, Rogers genetic distance estimation, and for UPGMA dendrograms. For the population structure analysis, we use the admixture model-based clustering using the software structure, and we ran principal component analysis in Tassel. We plotted the results using structure plot and ITOL. So how did we filter the SNP, the genotype data we received from Intertech Australia, from the panel? We first discarded any inbred line that had heterozygosity higher than 15%, and lines that had a call rate for a SNP below 50%. For SNPs, we only kept those who had a minimum allele frequency higher than 0.05 and a call rate of 80%. By doing so, we ended up with a matrix of data that contained 363 inbred lines and 2,187 genome-wide SNP markers. The density plot on the right, and I forgot to select the, let me see if I can do it now, the laser pointer. Yes, the density plot you can see now reflects the position of these resulting 2,187 genome-wide markers. I can't really see my full screen, but when we analyze the matrix, the genotypic matrix, and the data that the SNPs were providing us, we could confirm that we had a high proportion of informative SNPs. with polymorphic information content values on average of 0.32 and 90% of them being higher than 0.3. Minimum allele frequencies, the average across the 2000 SNPs was 0.32. And as you can see in the frequency distribution, many of them, half of them are really high frequency values. The missing data was across an average missing data for the SNPs was 2.55%. When we looked at the 363 invert lines in the panel, we could see that with the genotype, the marker data, we could see that they exhibit low levels of heterozygosity and are genetically diverse. Basically looking at the average observed heterozygosity, the average of observed heterozygosity was 0.02, and the inbreeding coefficient was 0.96. So basically, that indicates that the lines were pretty homozygous, and the genetic diversity estimates showed an average genetic diversity of 0.4. We also looked at the linkage disequilibrium, genome-wide linkage disequilibrium. I mean, genome-wide LDDK distance in this line, in this panel, was determined to be 154.5 kb at R² 0.1, which means that I mean, and we could see that there's a rapid LDDK as it is expected in mice, and this distance is within the level we were expecting to find. It's been seen that LDDK distances are 10 times higher in temperate than in tropical germplasm. We also took a look at the pairwise relative kinship distribution for these 363 lines, and what we could see is that most of the lines in the panel are either not related or only weakly related to each other, with only just a few lines showing strong similarity among them. Then we moved into, I'm sorry, I'm gonna drink some water. We moved into the analysis of the population structure. And we we use a structure for estimating the number of possible subpopulations within the panel. We obtained a delta K peak at K value 10, indicating that this panel could potentially be subdivided in 10 clusters. We also found a second peak at K value 2, and we went to first take a look at that clustering, and we could see that the line, 285 lines clustered in group 1 and 77 lines clustered in group, or had a membership in group 2. We also did an UPGMA tendogram to see whether this clustering hold, and we could see that it doesn't quite explain the diversity of the lines. When we looked at the when we did a principal components analysis, here the lines are painted taking into account the grouping obtained by the clustering obtained by a structure. You couldn't really separate the two subpopulations. Couldn't visualize the two subpopulations. Something that we could trying to find a pedigree explanation for these two groups. We could only see that most of the lines on the second group were dent or semi-dent, and the biggest group corresponded to the flint or semi-flint lines. So basically, we consider that a population structure of 10 subpopulation was most likely to be the one explaining the structure of this panel. And with a threshold level, with a low really threshold level of Q bigger than 0.5, 98 inbred lines were considered as admixture, so couldn't be assigned to any of these 10 groups. Fixation indexes, FSTs, values indicate highly differentiated populations. Also, there is a structuring in the population, except for cluster 9. Cluster 9 didn't show a high value of FST. I'm going to just describe you briefly which types of lines were how would you say it, with oh my gosh, I can't say it. Which lines belong to each of these clusters. So the first group contains 17 inbred lines derived from Cargill 280, mainly orange semi-flint type of seeds. The second group we called semi-dense synthetics, and that group contained or is formed by 10 inbred lines, like I said, semi-dense synthetics and lines derived from pioneer hybrids. The third group is formed by 15 inbred lines derived from an orange flint landrace that was very important that is very important, or was very important in the breeding program. A big group, a big fourth group that we called orange Argentine flints, that really grouped Argentine flints from different origins. A fifth group, B100, formed by 8 inbred lines, one of them being B100, and lives derived from crosses of B100 and a commercial hybrid. A sixth group with 23 inbred lines, mainly derived from one inbred line released by the program, LP 561, which is a red flint with resistance to mal de Rio Cuarto. A seventh group with 32 inbred lines derived from a synthetic population from the Iowa Steve stock heterotic group. Another crosses of B110 and commercial hybrid. So basically dent lines. Another group of 22 inbred lines with the well-known B73 in it, and other lines derived from commercial hybrids, which probably have B73 background in them. A ninth group, which is the one that this really did not have a strong support from the analysis. 11 inbred lines were assigned to that group, and with not a clear common origin. Some are red flints with a short cycle, so that's why we name it SCR. And a final group, number 10, which is formed by lines, 12 inbred lines from Argentina and lines from Argentina and Canada synthetics. I mean, from synthetic populations made combining Argentinian and Canadian materials. When we try to illustrate the variability, I mean the variability or the of these lines using principal component analysis, this only explains close to 11% of the variation in this panel explained by the markers. We couldn't really, I mean, not we, the analysis couldn't really show or illustrate the group, separate these groups. But what we could, what I could tell you is that, let me see if it appears, but it doesn't seem to be appearing. Okay, well, something's happening with the slide. Oh, no. We could see that on one edge of the graph of the plot, we find B7, which what I think it's interesting is that we find B73 on one end and LP 2542, which is a yellow dent line that was used as a tester. I mean, yeah, as a tester throughout several years when improving flint lines. On the other side, we find P465, which is a landrace that gave origin to many, I mean, contributed a lot with genes to this breeding program, and LP 612, which is one of the lines that has been used as a flint, I mean, it's a flint line that has been used as a tester for combining ability with dent lines. The dendrogram, if we paint the dendrogram with the colors resulting from the clustering with a structure, we can try to see that there's somewhat of a consistency in the grouping, having the P 465 group on one end, and also consistent with the PCA result with analysis we saw before. One end of the spectrum, the real Argentine land race and derived lines, other flint, orange flint, and the Argentine flints here. Here, this is a big, and like I said, a group with different origins that we could not discriminate with this panel of SNPs. The Canadian and Argentine Canadian synthetics group, lines derived from B100, lines derived from a Cargill line, Cargill, yeah, cultivar, synthetics, semi-dense synthetics, and the stiff stock, the more B73-like, and the more, oh, I don't remember now the background, B100, BS13, I'm sorry. So here we have the dense, more like groups, different groups with origins from different companies. And this one had some pioneer also germoplasm in it. I believe the Argentine and Canadian had also some materials from other Argentine materials, and this is basically the structure of this, the groups and the structure of this population that is somewhat consistent with the results from a structure. The ninth group, which didn't hold, is all spread in this dendrogram. So basically, it's not a consistent group. So mainly from the 10, we probably have 10 strong groups in this panel. To conclude, I would like to tell you that obviously implementation of this mid-density panel has allowed us to evaluate the genetic diversity of a big panel for which we didn't have marker information, complete marker information. We could confirm its high genetic diversity and complex population structure. Most of the inbred lines, like I said, are not related or are only weakly related to each other. These results for us really constitute a first step towards a better understanding and the reorganization of the existing germoplasm. You know, breeders retire and things, you know, need to, new breeders come in, and I think it's good for us to reorganize and have facilitate breeding decisions with this marker information. And also this study paves the way for genomic assisted breeding. We could very soon use it for marker assisted back crossing, and like I told you before, our idea is to hopefully start developing genomic prediction models, and many researchers in the group will start to phenotype and work on the particular traits they are interested in, and we will be able to make really good use of the the genotype data that we obtain using the excellence in breeding mid-density SNP panel. Well, with this, I would like to thank you, all of you, for listening, bearing with me during this presentation. Special thanks to Ana Luisa for her continuous support. And I want to make this acknowledgement. I mean, these results obviously are part of joint research that has been performed by the maize breeding group. And also want to thank my colleagues at the biotechnology lab who run many of the all of the microsatellite markers. And special thanks to Ignacio Torrent, who's working on the panel and who's doing his PhD. And I also, last but not least, I want to thank Agustin Baricala, who helps me with bioinformatics analysis, and two of PhD students who work with me and are always willing to learn and help me along the way. Thank you.
Thank you very much, Maria Laura, for the excellent presentation. I know that it's not easy to speak English when we speak every day Swedish. So thank you very much, really, for the effort. And we open now the floor for questions. And one of the questions that came was what is the selection criteria of SNPs to be used in the genetic diversity studies of maize? Are there databases where one can retrieve the sequences? So please kindly see my answer. So this is from our side, the answer. And Manje,
oh, okay.
Manje, you are here. If you want to unmute yourself and maybe you can explain very briefly. Otherwise, please see the link that I put here for the mid-density genotyping services. And people can read how the colleagues did from CMIT and IITA and other groups. And if you have any questions, can contact us. So I also open the floor for questions. And if you don't have any by now, I have one for Dr. Rodomiro and one for Maria Laura. So for Dr. Rodomiro, I would like to know what is your vision in what is most urgent matter in plant breeding that we need to address currently. I know that is very general, but what is your opinion?
No, that's the question for the how much was the question in the US? I think was in Peru was called the 64,000 soles question. That was a contest, like asking those questions and trying to figure out what could be a good answer. But let's face two things. First, we continue increasing population. In every day vary, but let's say between 800 million to 1 billion people go with hunger to bed. That means that we need to provide them with a missing food. At the same time, all of us, I suppose, agree that we are under global warming, climate change, so we need to produce more under this scenario, and on the top, do it in a way that will not be criticized because we need to employ sustainable methods. So there are crops on those three fronts. So you ask me what breeding can do, and I hope that this is the reason of your question is, yes, we need to breed for climate resilience, resource efficient crops, so we're producing more with less, that will provide the food and nutrition security to people in the world, particularly those that today are under hunger. It sounds like a propaganda that is doable. I think when Burlock did the Green Revolution, was more or less with a different background, but very similar. And they were able to do it. I think that today, with more knowledge, better methods and tools, we should be able to be up to the challenge. Is that answering your questions, Ana Luisa?
Yes, thank you. Thank you very much. My next question is for Laura. So what is the next step after this in your research?
Okay, I need to unmute. Well, basically, we first want to try to finish the genotyping, include all the lines we've been evaluating, and then start evaluating the panel for different traits. We have different breeders who work with different, specialize on different traits. So we could look at, for instance, cold response, abiotic stresses, like also the synchronization of the flowering. We have that type of problem here with the flints. The flowering is not synchronized, and when there's a stress, that gets worse. So all those, we do work with those traits, but maybe it's not, we haven't done it in such a big panel. So that's the idea, to try to unite efforts to be unite efforts and phenotype the whole panel for all the important traits we're looking at. Also, different phenological aspects, and obviously disease resistance is an important one, and the ER rods are important as well. So all of those traits, all of those breeding objectives are on the table. And the idea is to slowly implement genomics, and it'll be it'll be a step by step, but it's hard financially. Sometimes you don't get to have the numbers for doing big a decent analysis. So we're trying to unite efforts so that we can do that.
Thank you. Maria, you have here a question from Esperance that says, on which basis did you conclude that the population is quite heterozygote, despite the low level of observed heterozygosity?
Oh, did I say that? Because I didn't want to. Maybe I wanted to say homozygosity and I said heterozygosity. I don't know. The lines are pretty homozygous.
Okay.
Yeah. Maybe
Esperance, I'm
the
not
opposite.
sure if this answers your question. Yes, Cesar, please unmute yourself.
Yes, thank you, Ana. Yes, based on that question about the homozygosity and heterozygosity, I saw that you guys started from 484 inbred lines, and based on the heterozygosity, you eliminate almost 120.
No, I'm sorry. We sent only for genotyping 376.
Okay,
And
all right.
yeah, 4 plates. I'm sorry, that wasn't clear.
Okay.
Yeah. So only only a few lines were discarded. Some of them were discarded because they didn't have enough SNP data, like we have to repeat those, and only 1 or 2 had heterozygosity problems.
All right. Yeah,
3
that makes
maybe.
sense. That
Yeah,
makes
yeah.
sense.
I'm sorry, it wasn't
Excellent.
clear.
No worries. No worries. Okay. And
Thank you.
then you mentioned that the temperate lines they have more or less 10 times the LD of the temperate lines are 10 times bigger than the tropical maize germplants. That means that the temperate lines are more, let's say, homozygous compared with the tropical lines, and that's a good reason to to use this kind of technologies to make this breeding process.
Yeah.
Because we have a lot of diversity in the tropical and subtropical lines.
That's the thing, that it's a diversity difference among the pools, I guess. I'm new in this and I'm reading, so that's from the literature, and I was trying to see if we were finding a distance that made sense, and we are within what's being published. Obviously, it depends on the number of lines and the number of markers you use, but we're within the range, I would say. You're more of an expert, so you would know better than me.
And yeah, last one is to just let you know that in CIMMYT we have genotyped the whole germplants bank. So we have genotypic data, public genotypic data that we can just share with you, especially from the CMLs, the CIMMYT maize lines.
I know that panel is
lot
available.
Lot of them are, for example, resistant for common rust and many different
that'll be nice
diseases.
to combine and see what's we should talk. That's really it's nice to show the results with you guys, and hopefully create some
collaboration. Yes, sure. No worries. Yes, for sure. All right, that's all from me. Thanks, Ana.
Thank you, Cesar.
Thank you, Maria.
So if nobody else have any other question, I think we can go to a conclusion. So I would need to take a great amount of time to say thank you to all the colleagues that participate in all these series of seminars. But I just want to really say thank you very much to all. Thank you, Maria. Thank you, Dr. Rodomiro, for your participation and your time. Thank you to all the speakers that had also the time to participate, almost 1 year. The seminars presented here are just a small sample of the CTH project and about what we do and how we can support as a CGIAR, NARS and NARIS around the world, but particularly Africa. So ranging from maize, wheat, sorghum, banana, yam, and other crops. We also support many partners on other crops, such as groundnut and rice. From Africa, Asia, and Latin America, we also assist partners in North America and Europe. But this is not done alone. So partnerships are made with vendors, such as Intertech and diversity arrays that are crucial to achieve this task. No less important are the support made from communications, colleagues from the CGIAR, ICRAF, and CIMMYT in supporting in the flyers and arrangements. Finally, a note of thanks for the donors for the continuous support and in our activities. So all of you are extremely appreciated. Thank you to all. Thank you very much.